Good morning, you three.
Good morning.
Good morning.
Oh, hey, Michael. Missed you last time.
Yeah, I apologize for not being here. I forgot that you already created the link, and I thought you were going to email one out, and I missed the last two. But
No,
I'm here.
it's the same link for all of them, as you probably already know, right?
Yes, now I do.
Okay. Well, no worries. The videos are up, so you'll be able to kind of follow through what we covered last Thursday and Tuesday. So hopefully you shouldn't be too disadvantaged from that. Let's see. So what we're going to do this time round is finish off translation. Where are we at? Translation. Do you want to do chemosynthesis? Lots of fun. I think we're at here. Oh, hey, Erica.
Hello.
So basically we finished up on Tuesday. I can't remember what day. It's not Friday. I thought it was Friday. It's not, it's Thursday, obviously. So ignore my chat greasing. That's obviously wrong. Let's get rid of this before it pops up and annoys me again. So yeah, anyway, so we finished up on Tuesday with translation termination, right? So it's much simpler than transcription termination, which has a whole bunch of varied mechanisms depending on species and transcript. It's simply a release factor, right, that recognizes the space left by that stop codon, because there are no tRNAs that recognize the stop codon, and it comes in and pushes the ribosome off of the RNA. So a couple of little extra bits, right, which aren't what I would call central to the whole process, but interesting wrinkles that are worth knowing, is that this isn't a case of one ribosome per RNA. As soon as enough of the RNA has been kind of extruded from the ribosome, let's say, how much is that? About 25 amino acids. So that's the 5' UTR plus about 75 bases, give or take. So it can be a relatively short amount of mRNA in the, couple of hundred or so bases. But as soon as that, let's see where we're at. Here we go. So this is for eukaryotes on slide 20 right now. Pardon me. As soon as that ribosome is scanned along and produced about 25 amino acids, so it's going to be over here by some distance, another ribosome can start the whole process again, right? Can assemble on that 5' UTR. Same process, exactly the same concept. You know, the small subunit binds to the cap proteins, and methionine tRNA comes in, scans, starts, and over and over again. So for proteins that need to be expressed at very high levels very quickly, let's say, you know, you've just had a large meal and your blood sugar's going up, and so your beta cells in your pancreas are dumping insulin into your bloodstream, right, to reduce that blood sugar concentration. As soon as that's done, your beta cell is going to need to produce a lot of insulin very quickly to restock, so it's ready for the next blood sugar peak. And so for genes that need to express at high level, not only can you produce a lot of mRNA, you can also produce a lot of protein, and these things are called polysomes. And so essentially they get the idea with, it's like a production line, right, going from top left to bottom right. Ribosomes assemble, start producing amino acid, move along far enough, another one assembles, and each one kind of goes along and it's, the same rate. And so you have this kind of chain of ribosomes on the mRNA producing amino acid, right? So it's a way of very quickly producing protein. And do we have, yeah, Rose is here. So Rose, this is also another good example. Oh, hi, Simon. Of why you want to control how long that mRNA hangs around for. So if you only want a specific amount of protein, or for protein for a certain amount of time, you need to get rid of that mRNA pretty quickly, because otherwise it will just keep producing protein over and over and over and over and over again until you have more than you need, or you've wasted resources on making something that's not necessary. So getting rid of that mRNA, typically via degradation in the 3' UTR, which is kind of down here past that UAA, that stop codon, is pretty central to controlling gene expression. Right, so it's just an interesting example of that. The other wrinkle that's worth going over quickly is that one of the main differences between gene expression in prokaryotes and eukaryotes is that in prokaryotes, those two things, gene transcription and translation, are linked, right? And this is, I would strongly recommend like your ears pricking up at this, because this is a really important way in which prokaryotes control gene expression, right? Because those two things are linked, you can actually use structures in the mRNA to control whether or not that mRNA gets translated, right? And that's really the foundation of what we're going to learn, or start learning, next week after the exam, in terms of control of gene expression in prokaryotes, right? So it's a really important aspect of their biology. Let's stick that a little further over. Oh, hang on, I'm kind of jumping around a little bit here. I'll go back to cistrons in a little bit. So essentially, because prokaryotes don't have a nucleus, as soon as that mRNA, pardon me, starts being transcribed and the initiation site, the Shine-Dalgarno site, becomes available, a ribosome can bind to it and start translating it. So they have polysomes like we saw up here, right, as we have in we have too in eukaryotes. But they can make that protein while the mRNA is still being produced, right? So they're very tightly coupled. As soon as the mRNA emerges from the RNA polymerase, ribosomes can bind to it and start producing amino acid, right? So it's a really effective way of both producing stuff quickly and also controlling gene expression too, because we'll get to that next week. It's not easy to give you a quick summary. Essentially, the structures in the RNA or the mRNA tie into that translation to either promote or inhibit translation. Another big difference between prokaryotes and eukaryotes is prokaryotes tend to group all of their protein coding regions in a single kind of regulatory unit. Tripoperon is a classic one, lac-operon, others as well. So if you want to do a thing, whatever that is in bacterial terms, let's say take up and metabolize a sugar, right, lac-operon, for example, instead of using glucose. So to do that, you need to do a lot of different things, right? You can't just produce an enzyme that turns lactose into something useful. You also need the enzyme or the proteins that allow that lactose to be taken up from the environment. There's a bunch of different things that need to be made to do that one job. And so in prokaryotes, they stick those in what are called operons, which is O-P-E-R-E-N. Let's just write that down there. Operon. There you go. That's a good definition as any. And so when that mRNA gets produced, then within that single mRNA, you have what are called multiple cistrons, right? Which are regions that code for protein. And so they're called polycistronic, right? So essentially you have all your genes grouped together in what's called an operon. They're all under the same control, right? So you turn it all on at once. And then once you produce that mRNA, then you can make multiple proteins, different proteins from that one RNA, right? Which is very, very different from eukaryotes. Eukaryotes just encode one protein. Eukaryote mRNA, sorry, only encode one protein, and that's it. On the flip side, which is also what we'll get to when we talk about control of gene expression, instead of coordinating gene expression by grouping all the same things and all involved in the same process together, instead of doing that and put it under a single unified control in a sense, instead what eukaryotes do is that they have these genes that are dotted around throughout the genome, but their expression is coordinated by transcription factors, other proteins that recognize the promoters of those genes. So one of the examples I always use is a heat shock response. You know, if you start heating up, your proteins start denaturing, you know, it's generally bad news. And there are lots of different aspects of the heat shock response, lots of different proteins produced. And so each one of those genes in its promoter has a bit of DNA that's recognized by a heat shock transcription factor. So you can coordinate expression over all of those different genes in a similar kind of way that, or to the similar end that prokaryotes do, but they're a very different mechanism. So yeah, those are two big ways in which prokaryotes are different from eukaryotes. One is the tight coupling of translation and transcription. Two is grouping different protein coding regions together in one, regulatory unit, and then controlling that all at the same time, essentially. So those are the big differences between the two. And obviously that's going to have a big consequence in terms of how the gene expression is controlled as well. Does anybody have any questions about any of that stuff before we move on? Let me make sure I have the, don't want that. Come on. It's always a struggle. There we go. You can
So with the,
oh, hello.
sorry.
No worries. I was just going to say, if you want to write up questions as they occur to you, you can stick them in the chat box.
So since transcription and translation are linked in prokaryotes,
Mhm.
that would make them more efficient in like producing these proteins and things that they use for function.
Yeah, they're able to produce those proteins much more quickly. So in kind of simple terms, they can make a lot of protein much more quickly than eukaryotes can. On the flip side, they have less fine-grained control over how those proteins are produced. And also they're able to do less things with those proteins in terms of processing than eukaryotes are. So there are definite pluses to being able to not having a nucleus, essentially, right? in terms of gene expression, but there are a lot of minuses as well. So it's kind of more efficient in one sense and way less efficient in another sense because you know, they don't have organelles, so you know, they kind of suck. Which is funny because I did my degree was in microbiology, and I'm a eukaryote, you know, for the win kind of scientist now. So yeah, it's just a different way of doing things, and it's more, I'm gonna guess one could think of it in terms of it's more suited to the physiology of that organism, of prokaryotes. So prokaryotes need to be able to quickly respond to changes in environment, you know, in terms of food availability or food type, you know, various things like that. Whereas eukaryotes, particularly multicellular organisms, need a much more nuanced response, right? It's much more complex, essentially, what we need to do and, you know, how and why. So this kind of approach really wouldn't work in eukaryotes for a bunch of different reasons. Whereas in prokaryotes, you know, it's like, you know, they don't think, they go, this environment sucks, right? Let's kick on the this environment sucks packet, and then they'll just stick it all on, and then they'll be able to cope with that fairly quickly. Whereas eukaryotes they have much more control over their environment or where they are in their environment than prokaryotes do, and what resources they use, and they're just a lot more complex. So this kind of simple, almost like Soviet style approach works for fairly simple kind of communal organisms like prokaryotes, doesn't necessarily work as well for eukaryotes. So it's not so much that one is better than the other, it's more that one approach is better suited, or the approach is suited to the organism, essentially, I think is the best way of thinking about it.
Okay, that makes sense, because I was thinking also like it has to do with the complexity of the cell. Like prokaryotes, they don't have the organelles, so they don't have many options when it comes to.
I think for prokaryotes, it's more for eukaryotes, because so essentially in prokaryotes you have to do everything in one room, right? Basically. So you're storing your DNA, you're transcribing it, you're translating the mRNA, you're doing your enzymatic reactions, your metabolism, and so on and so forth. And so it's kind of like a caveman analogy, right? You know, it's like you're living in the cave. Cave's good, right? If you don't have a cave, at least. But I'd much rather have, you know, a 3 or 4 bedroom house with a living room, a kitchen, a bathroom, right? You know, not crapping outside in the woods kind of thing. And so organelles are far more efficient and allow you more options, right? So in terms of eukaryotes, there's a lot more control over the whole process from DNA to functional protein, you know, and there's a lot more after proteins are produced. So if you remember us talking in GenBio 1, for example, about export or transmembrane proteins, right? Processing. There's a lot of things you can do to those proteins to control their activity and their function that prokaryotes can't. And so in prokaryotes are much simpler, right? They need this done, they get it done. All of this stuff, they produce it all at the same time. Eukaryotes are much more complex, and it's like, well, we need some of this, but we also need some of this, but we don't need that yet. And this needs to go there, and that needs to go here, and we could really do with some of this as well later, right? And so there's a lot more kind of fine-grained control over a particular response or a particular process than there is in prokaryotes. So it's very much the, you know, within the context of the organism, right? It makes more sense.
Okay, thank you.
And eukaryotes are better. No, that's silly. You know, we would totally suck at living in a nuclear reactor, whereas some bacteria are totally fine with it. It's just we suit the niche that we exist in. Our niche just happens to be very large, but bacteria are also pretty amazing at living in particular niches too. Oh, quick aside, I was for my astrobiology. Do I actually still have that open? Here we go. Don't know if you can see this. I should do. This is my favorite picture of Arnold Schwarzenegger. Anyway, there's this bacteria here. Actually, yeah, it's a bacterium. It's basically a radiation-resistant cute berry. That's what its name means. It can resist about, well, it can live out, sorry, about 1,000 times more radiation that would kill us within 2 weeks. So that's in grays. So, you know, expose us to 5 grays, which would be kind of like going in and working in the reactor room of Chernobyl just after it exploded for a couple of days. That would kill you, right, within a couple of weeks to a month. This bacterium can resist over 1,000 times, up to 3,000 times the amount of radiation. So, you know, the bacteria are also pretty cool as well. Let's get back. And it means I can use a Conan the Barbarian picture too, which is always fun. Hope you appreciate that. Any other questions on any of this stuff?
I had a question. So for the polysome,
Yep.
that only is needed like when you need mass amounts of the same type of protein, or is it?
Well, it will occur anyway, simply because ribosomes, like RNA polymerase, they're dumb things. They're just molecular machines. And so they don't know in and of themselves how much protein they should be producing. They see a 5' UTR and they'll bind to it and they'll make some protein. So there's other stuff, actually not circling here is no good because that's the start of the coding sequence. There's other sequences in the 5' UTR and 3' UTR that control how efficiently ribosomes find and bind the 5' UTR. At least I'm not so sure about prokaryotes. I don't think that's really quite true. I'm not really very sure. I know a lot less about prokaryote gene expression than I do eukaryote. But in eukaryotes, at least, you can control how many of these or how frequently these ribosomes kind of load up and translate that mRNA. So in answer to your question, yes, that is typically when you need a lot of protein. If you need less protein, say kind of at a more steady rate, like for what are called housekeeping genes, for example, that produce ribosomes, right? There would be something in both the 5' and 3' UTR that would essentially, get off of that. That would essentially affect how efficiently that ribosome binds to the 5' UTR and then starts translating the mRNA. So you can control the amount of protein also by how often ribosomes are able to translate it, which is kind of crazy. So hopefully what you're getting from all of this is that there are an astonishing number of ways in which you can control the end product in terms of gene expression. And so you're right, how many ribosomes you have translating the mRNA can also be dependent on the mRNA being translated and how much of that protein you need. Yeah, so there's a lot of In fact, there's so much going on in terms of gene expression. It's its own field in science. It's called gene reg, and it's astonishingly complicated. Like, you know, kind of when I look at that field, it's like staring over the abyss. You know, I don't want to go there because I'll just get lost and I'll never find my way out again. Right? It's just that astonishingly complicated. The deeper you go, the more complicated it gets. And so, yeah, there's an awful lot of stuff that can go on, and that's just one of them. Hope that answered your question.
Yes, yes, it did. Thank you.
Cool. Who knows, you may end up going on to study gene regulation. Huh? That'd be funny. And you'd be like, my genetics professor, he is all scared of this kind of stuff. Anyway. Cool. All right. Pretty much all we've got left to do on this one is just to go over again this idea of reading frame and degeneracy. Right, so you get on that. So the reading frame is essentially the series of three-letter words, codons, right? That always starts with AUG. Let's see where we are on here. Oops. This is what we want. So if you can see here on the bottom of this image, here's our AUG, that's the start codon, that starts the frame, right? The next word is 3 letters past that. The next word is 3 letters past that, and so on, and so on, and so on, right? And so this whole mRNA is read in a series of words, right? Those words called codons, right? And the frame, the pacing of those words is solely dependent on that start codon. It's like if I gave you a sheet of letters, right? You know, like an essay, and just removed all punctuation and spaces. And essentially the first word in that, you know, we could, as a kind of cutesy example here, right? The big boy, big boy saw the new cat eat the hot dog, right? Seems a little silly, right? The reading frame is solely provided by that first word. That's it. The is it. And then every other word is read in reference to that first word in that sentence, right? And so that's the reading frame. And because it's dependent on that first word, if you add or subtract letters from that, right? So if you take away B here, for example, you then shift that reading frame, right? This is probably a better example here, right? This is an insertion of 1 letter, right? And so essentially now you have, I don't know, kind of pointing at it, you can see that the original reading frame has been shifted. But because the frame is in reference to the first word, you got 3, 3, 3, 3. This is now the next word, right? So it's TTH, and then ENE, and then WCA, and TAT. There you go. Should recognize that as a Brit. And you get the same thing if you take away a letter, or 2 letters, or add 2 letters, right? However, if you add 3 letters, where are we at? There, where's this? Well, they didn't do an addition of 3. Anyway, if you add 3 letters, then you add 1 new word, but you don't change the words after that new word, right? So you essentially you add another amino acid, but you don't change the meaning of or the identity of the subsequent amino acids, right? And so that's really, really important in terms of point mutations, right? Deletions, insertions, things like that, because each of these words is recognized by a tRNA. That's called degeneracy, right? So essentially it means that you can have 4 different words. There's actually one that has 6 in here. Never mind. Oh, here it is, leucine. Right, you can have 6 different words which mean the same thing for leucine. Right, you could have UUA, UUG, CUU, CUC, CUA, CUG, right? So in terms of leucine, the most important letter in that word is U, the second one, right? And the next most important one is the first letter. Typically, it's the first 2 that really matter. The last one generally matters very little. It sometimes does. You know, so you can see here in this box up here, if you change the last letter in these words from a C to an A, for example, you'll go from a phenylalanine to a leucine, right? So you do have a missense mutation, right? A change in the amino acid sequence. If you go from a C to an A in these words at the 3rd position, it doesn't make any difference, right? So there's essentially like multiple redundancy in terms of the words that are used to write amino acids. And that also means that this image here isn't actually a very good analogy, right? It's kind of handy to think about it in terms of reading frame, but it really doesn't work in terms of degeneracy, right? And so an example would be that you can have 4 different words that both mean cat, that begin with CA, right? So you could have cat, cag, cac, and car, right? All mean cat, right? So that's kind of, that would be a better, and so you could change the last letter of cat, you know, to anything else and it'll still mean cat, right? You know, that's kind of one way of thinking about it. And then there's a little bit about wobble, which is basically that the last, some tRNAs can come in and bind even if their last letter doesn't quite match. And the reason for that is that the first 2 letters are really the most important ones. Doesn't always apply, because obviously you wouldn't want that happening, let's say, with tRNAs for serine or arginine, right? Because you'd end up with the wrong amino acid in there. But for some of them you can. Right? And so that genetic code is universal. Some species vary in which of those words they use to encode a particular amino acid, right? It's not uniform, and that also depends on, you know, GC content and things like that of the DNA. But these words are all the same across all species, right? So this is a deeply ancestral system, right, which occurred almost certainly soon after life first emerged, right, and started producing proteins, right, which was, you know, probably the 3rd or 4th thing that life decided to figure out how to do, because this is shared by all life, right? And it's not just all life now, it's all life that has ever been, as far as we can tell at least, uses this genetic code. Okay, so that's pretty much it from me. The rest of the session is really up to all of you. Any questions that you may have about this stuff or about any material since, oh, seems so long ago. Where was it? Since spring break, basically. So the exam next Tuesday will be on all of this stuff. Eukaryotic structure and organization, transposons, basics of gene expression, transcription, initiation, elongation, termination, right? Really important. RNA processing in eukaryotes and translation in prokaryotes and eukaryotes, right? That's what exam 3 down there is going to be on. So if you got any questions about any of that, now's the time to ask. It also means I can have some of my coffee. And hopefully Alana is going to ask some questions so I can hear a dog snoring too. Still got about like a third of a cup, so have a think. Let's see if anyone's asking questions on chat. Who else do we have? Oh, John's here. Hello, John. Esmeralda too. All right. Anybody have questions? Questions? Come on, keep me fed. Otherwise, I'm just going to start reading the news, and that's all kinds of depressing.
I have a question about
Fabulous.
the Shine Dalgarno sequence. So I know that it binds to an mRNA ribosome along with like some energy, binds to
oh yeah, here we go.
whatever it binds to. But like what is it?
It's a sequence in the mRNA in the 5' UTR of prokaryotes. So let's go even further back because I want to. Where is that? It's around here somewhere. Here we go. Okay, so for example, right, so this is going way back. This is now transcription, right? So there are in both prokaryote and eukaryote promoters these things called consensus sequences. These are bits of DNA, like a particular sequence or like an approximation of a sequence, right? They don't have to be exact. But they have to have like similar kind of properties, essentially, that proteins bind. And the same is the case in eukaryotes, actually way more so in eukaryotes. And so these proteins recognize, remember how DNA is like a double helix. They recognize a particular part and kind of bind to it, right, via their own inherent structure. And so for transcription initiation, those sequences are bound by RNA polymerase or Tadalbox binding protein in eukaryotes, various proteins, transcription factors, et cetera, et cetera. So in prokaryotes in translation, that Shine-Dalgarno sequence is the same kind of deal. It's a conserved sequence of bases in mRNA in the 5' UTR, so this is before the start codon, that is recognized by the small subunit of the ribosome. And so essentially there's, if you can see where my cursor is on screen, there's part of that small subunit, say around here, that quite closely fits DNA of RNA of a very specific sequence, that Shine-Dalgarno sequence. And it's called Shine-Dalgarno because those are the people that discovered it, basically. And then it will bind to that. So this small subunit is not going to bind anywhere else on that mRNA, because you don't want it binding in the middle. You don't want it binding at the end. You want it binding right very specifically at the start. And that Shine-Dalgarno sequence, you will only find it a very specific distance upstream or ahead of the start codon. It has to be, right? Because if you think about it, we've got, if you, right, let's just jump back here, use this as an example, right? So this is prokaryotes, but the principles are the same. that large subunit has 3 sites in it. The access site, the peptide bond or pairing site, and the exit site. So when that prokaryotic ribosome assembles, it has to do so that the start codon, the start tRNA, essentially, the methionine tRNA, is in the P site. And so if you have something of a certain size, it has to fit exactly like so. And so this Shine-Dalgarno sequence has to be a very specific distance from that start codon, so the small subunit can find it, right, and bind to it, and then the large subunit assembles on top of that with the start tRNA right in the middle. So that's what the Shine-Dalgarno sequence is. And it's also very specific to prokaryotes, because in eukaryotes, they don't have that. What they do instead is that small subunit starts at the very, very, very end, and then it scans along the mRNA until that tRNA recognizes the start codon, and then that's the start of translation, and then everything assembles on top of it after that. So it's 2 different mechanisms to get the same output. In bacteria, they get that ribosome in the right place by using a conserved consensus sequence just upstream or ahead of that start codon. In eukaryotes, they basically scan it like a piece of ticker tape or something like that. You know, you just kind of keep pulling it through until you find the word you want, right? And so they don't have to assemble the ribosome in the right place from the get-go. They simply attach it to the 5' cap, and then it wanders along the mRNA until it finds the start codon. Does that make sense?
Yes. Yes.
Yeah.
Thank you.
So it's something inherent in the mRNA, is essentially what that sequence is. It's a feature, right? Basically, in terms of DNA sequence or RNA sequence. Sorry, I was getting too confused. Rose, did you have a question?
Yes, I had a question on the PowerPoint 15. You were just on it, where the Tata box is, and it says promoter sequences in eukaryotes are generally much longer and more complex than those in prokaryotes. Is that just because the promoter regions in the eukaryotes have like 2 types of promoters, because it's the regulatory road promoter and the core promoter, or
it's because eukaryotic gene control of eukaryotic gene expression is far more complex than in prokaryotes.
Oh, okay.
So prokaryotes, they have a more conserved promoting. Get off of that. But they're a lot simpler in terms of how they are turned on and off. Right. Typically, it's through presence or absence of amino acids or inhibitors or activators. They're relatively simple, right? So you don't have that distinction between the core promoter and the regulatory promoter that you do in eukaryotes. So in eukaryotes, you have typically the bare basics that's required to turn on a gene to transcribe it. And then you have a potentially huge amount of DNA sequence which can be involved in modulating that. And when I say huge, I mean like millions of bases long, for example. So the reason why, have you ever seen Hemingway's cats, polydactyl cats? Let's see if I can find some for you. Even better.
Well, you have so many tabs open.
I've actually culled them. That's quite a Who doesn't like pictures of kittens? Look at that. They're super awesome. They're very, very efficient at killing things too, because they've just got such a massive paws. Anyway, better not get too deep into the weeds in terms of cat pictures, because, you know, whew, that would just go on forever. But let's bring my screen back up so I can see what I'm gesturing at. Here we go. So essentially, the reason why you have this many fingers and this finger is different from this thumb is when you were a wee tiny embryo with little limb buds, limb nuggins. There's a protein produced here called Sonic Hedgehog, right? After the computer game character, and that diffuses kind of across that limb bud. Basically, it looks a bit like that, right? You know, you don't actually have fingers yet. And so where there's a lot of it, you have pinky, a little bit less, you have whatever that finger's called, and so on and so forth, right? And none at all, you get a thumb and a forefinger. So the thing that controls that and stops it being expressed here is a small piece of DNA that's like, I think is around 1 million to 2 million base pairs away. So this would be the equivalent of, I don't know, Lafayette, right? Relative to like my house being the gene. That regulatory sequence would be all the way along the coast, you know, where they have po' boys and stuff. And so if you disrupt that small regulatory sequence, right, it's technically part of the regulatory promoter, even though there are, you know, hundreds of genes in between, right? So it's not contiguous. It's not like one continuous thing. Then essentially you end up getting extra fingers here, because that control of the expression of that gene is disrupted. And in cats, you get that. Isn't that so cool? Look at that. I wish I had a polydactyl cat. A friend of mine had 3 of them actually, but he never let me take pictures of them because he thought I was going to like experiment on them or something. There you go, more cat pictures. Big cat paw pictures. Look at those paws. And some more. Anyway, so you could spend all day looking at cat pictures. See? Look at those paws. It's kind of hard not to get too excited about it, to be honest. So yeah, largely to reflect how complex control of gene expression is in eukaryotes, whereas it's really relatively simple. It's not simple, as you'll find in a little bit, right? Because we'll be going on to that. Pardon me, but it is simpler than in eukaryotes for sure, and so that's the distinction between prokaryotic promoters, which are relatively, where are we at? Relatively simple, right? They're not, you know, totally simple, obviously, but compared with, so here you go. Yeah, you basically have just a limited number of what are called sigma factors coming in, whereas in eukaryotes you have a huge number of transcription factors and activators, enhancers, inhibitors, and so on and so forth, right? And so this, oops, where are we at? Yeah, go back again. This DNA looping round, right, which you can see here at the bottom of that figure, that bit of DNA can be extraordinarily large, right? It doesn't have to be right next to the gene that's being activated, that's going to be transcribed, right? It just has to have that small bit of sequence in that DNA that is recognized by that protein, and then that protein interacts with this transcription apparatus and then activates it essentially, or inhibits it, depending on what it's doing. Yeah, so it's a lot more complex.
Oh,
That's for sure. Alana, did you have a question? I saw your audio come on.
Yeah, I had a quick question in the slides 15 and then 1 and 16.
So which PowerPoint? 15?
So in 15, slide 21, the visualization for consensus sequences, I'm kind of having a hard time
Oh, okay.
with that.
So really all this is if you take, let's just focus on the -10 sequence, that Tartat box, right? Essentially what you do when you line up all these sequences, right? As many as you can, basically. And then you go, okay, how often is this base found at this position in this sequence, right? And so if you look at the last T of the Tartat sequence, every single one of those different genes, different promoters, has that T there, right? So for example, I don't have a T here, bugger it. But anyway, that would be one single letter, right? So all of the different sequences that are lined up, they all have that one letter there. So that's obviously a very important letter because it's present in all of them, right? That must be something that is absolutely required, because if you don't have it, then you don't have a functional gene anymore, right? Whereas if you look at, say, the next letter along, the A, actually you can have any letter there, right? It's not particularly, there doesn't seem to be any particular rhyme or reason, right? You've got T, A, C, T, T, G, C, A, right? So there you're much more likely to find, it would look a little bit more like one of these, right? So essentially the height of the letter, well, the letter present is what you find there. The height of the letter is how often you find it there, right? So this is really just a way of visualizing what letter you have and how often you find it. So if this were a consensus sequence, for example, you'd probably say that it was a CAG something ACC, right? And then a couple of bases of who knows what, and then G something CAG, let's say, right? So these letters like nucleotides 10 and 11 are almost certainly unimportant, right? Because there's no conserved base in that position. Whereas these two letters at 8 and 9, these two C's, have to be important, because otherwise over evolutionary time, there will be random mutations which will change those, right? And so the fact that those letters have not changed means that if they do, the thing stops working, right? And that's obviously bad for the organism, so it doesn't survive to pass on its genes. And so these particular positions, like the CC and the AG, are strongly conserved, probably because they're very important in whatever function that sequence has. And typically, this wasn't a very good example, but let's go back here, right? Typically, that would be part of that protein which recognizes that specific base, based on like an amino acid residue, side chain properties, whatever, right? Shape of the protein, perhaps a charge residue there that recognizes a T, let's say. So if that base is not present, that protein won't bind to it, and then the process doesn't work, right? So that's essentially what consensus sequences are about. They're like the bare minimum needed for function, and depending on what bases you have may affect how well or not like the degree to which that process works, but you have to have some of those, otherwise it doesn't work at all.
Okay.
So it's just a way of visualizing it, really. Oh, you had another question for a different PowerPoint.
Yes, I had a question in PowerPoint 16 on slide 22.
Yeah.
For, I guess, step 5 and 6. Well, 4, 5, and 6. It forms an IF2 and GTP complex.
Right.
And then binds to the initiation codon. How does it go from 5 to 6?
So essentially, I wouldn't get too far into the weeds with all of these different things, the
Okay.
various numbers of the initiation factors and stuff. It's more important to understand the general process. So in prokaryotes, if we just kind of go back up a little bit, the small subunit is what defines the start of translation. And really that's
Okay.
defined by the Shine-Dalgarno sequence, right? Again,
Mhm.
different from eukaryotes. So once that small subunit is bound to the mRNA in the correct position, basically you need to bring in a tRNA that's charged with methionine. And that's what these various doohickeys up here do, right? So the tRNA comes in, obviously recognized as the start codon, and it comes in with these initiation factors and GTP, right? So GTP is an analog of ATP. So it's a form of cellular energy, much less frequently kind of encountered, but really, really important, right? So, and it behaves in the same way. And so essentially this comes in and this is energy, right? And so when you want to assemble the large subunit on top of that start codon, mRNA, and small subunit complex, right? You need some energy to both get that stuff on and get these other things off. And that's what's provided by the GTP.
Okay.
See that GTP is then hydrolyzed to GDP and, phosphate.
Mm-hmm.
And that's what provides the energy to bring in the large subunit, stick everything together, and kick everything else off. I'm not 100% sure about exactly which bit does, does the GTP, is that involved in bringing in the large subunit or kicking off the other bits, which then allow the large subunit to join? I don't actually know. It's not really that important to know, to be honest.
Okay, I just wasn't sure because the first couple steps showed exactly what was going on, and then it got really generalized towards the end.
Right.
So I was just wondering how that worked.
Yeah. So really this initiation factor here prevents this from happening. Right.
Mhm.
So it prevents these two ribosome subunits from assembling when they're not on mRNA. Right. So that small subunit is kept apart. That allows it to bind the mRNA, and then the large subunit is kind of guided in by the presence of this tRNA with methionine on. And the reason for that is, if we go all the way up here, that methionine charged tRNA has to be in the P site. That's the same for actually both eukaryotes and prokaryotes. It has to be here because the second codon, that tRNA has to come in, and then you get a peptide bond formed between the methionine and then the second amino acid. And that's when translation gets going. Right? So you can't have the methionine tRNA be in the A site because, where's the next tRNA going to come in from? You have to move it one word along to be able to start the whole process. And so that's why for both prokaryotes and eukaryotes, this large subunit has to assemble on top of that mRNA, and basically your small subunit is kind of here, like so. That large subunit has to come on top with the tRNA, the first one, sticking right up in the middle in the P site. And that's really what's kind of done in detail here. So your tRNA is coming in because it recognizes the AUG. It's also helped by these other initiation factors. And then once that occurs, then the large subunit can then assemble on top of that with the help of some energy from GTP.
Okay, thank you.
Does anybody else have any other questions? Speak now, forever hold your peace.
I had another question, and it was on slide 9 of, I believe, PowerPoint 15.
Okay.
And that was just like on the directionality of the genes, like because the, I was just wondering what was the correct orientation of these complementary DNA strands? Because I know that the mRNA is transcribed in the 5 to 3' direction. So does that mean that the DNA strand is in the opposite direction?
This is an interesting question. It's a little bit funky to talk about. So the issue here, Rose, is that the strand being copied is not the strand that's being used as the template. Squeaky lids. Let's get rid of this for a second so I can bring this up larger. Okay, so yeah, move this over a little bit. Couches are really comfortable, but they're not necessarily the best places to teach genetics. Okay, so this is 5', 3', 5', 3', right? So that's your gene. Let's put it a little bit further back, actually. Sorry about the reflection from the window. Let's see if I can put a different light. Let's see if that's a little bit better. No, that still sucks. Let's do it over this way instead. Haha. Okay, so this is your gene. Let's say, oops. There's your gene there, right? That's the bit of information you want to copy. So what you're going to be doing is you're going to turn this. It's kind of, I'm not very good at writing upside down. You're going to turn this into mRNA, right? But to do so, you're going to be using the strand underneath it as the template, right? So when we talk about actual transcription, right, this strand here, the 5 to 3', the left to right one, that one's not actually being used by the RNA polymerase. It's the complementary strand underneath it, which is the template, the one that's going in the other direction. Right, so when you're actually looking at transcription, if you had a gene that's going left to right, you're actually using the other strand to produce that mRNA in that left to right position. That's why it looks kind of funky in the PowerPoint. Let's see if I can get back. Yeah, so if you look at, let's just bring it back up again. Right, so if you look at the, oops, here, the gene is going, yeah, so essentially the gene direction, let's say for this first transcript here, it's a little bit weird to get used to because it's kind of the other way around from what you think, right? Mm-hmm. For this very first one on the top left, the gene is actually on the bottom strand going right to left.
Oh.
Right? And yeah, and so the actual transcript is being made using the complementary DNA strand. So the actual transcription start site is on the other strand.
Oh.
Right? So if you, the one that's most analogous to the drawing that I put up here is the one in the middle. Right? Don't forget that obviously these are complementary, right? So you get the opposite bases in them. They're not different. They're just the opposite of each other. But the closest analogy, the one that's exactly like this, is this second gene in the middle. Right, so they're the gene sequence that's being copied, right, into mRNA, is on this top strand going left to right. Mm-hmm. And so transcription actually occurs on the bottom strand, the opposite strand, because it has to, right? The only way that you get the right pairing to go from 5' to 3' is if you use the complementary strand as your template. Oh, okay. Just as you would for DNA replication, right? And so this is also a neat kind of example of you can actually have two genes which are controlled by the same promoter, right? So if you have a gene like in the slide 9 on the PowerPoint 15, if you have another gene that's kind of, say over here, right, in this sequence, going that way. Let's make sure you can actually see that. This region here, this is going to potentially be a shared promoter. Oh, okay. So it's going to control the expression of both this gene going this way and that gene going that way. Right, but when it does, the transcription of this gene, the one on the bottom strand, is going to be off of the upper strand because it's going in that direction. So this is going to be 5' to 3' that way. It's just a little bit weird getting used to that idea. You know, the fact that the strand being used is not the strand which contains the information in a way. I mean, it kind of does, obviously, but if you read from left to right, really you're going to be writing that like using the complementary strand going in the other direction. Yeah, it's kind of weird to try and explain, but it's really just based on the fact that DNA is an anti-parallel deal, and you can only make nucleic acids in one direction. I got a, oops. Oh, Alana, you need to go. No worries, Alana. Any questions, just send me them by email and I'll answer them before over the weekend at the latest, okay?
Alrighty, thank you. Have a great day.
No worries. Okay, everybody, I've got to get off because I've got something I need to go to as well. Hey, love, you got a coffee in the pot, sweetheart? Again, if you got any other questions, let me know. If Michael and anyone else who wasn't able to attend the last few video sessions, check those out, because essentially that's where we go through particular things in more detail, right? So stuff which you know, is of particular importance. So those are well worth spending the time going through, obviously in addition to your own revising and stuff. All right, everybody, I gotta go.
Thank you.
Have a great weekend, everybody, and I'll send out more information about the exam once I've finalized the details. I've kind of given you some general ideas so far, but I'll know exactly what's going on once I've actually done it, which will be hopefully by the end of this week. Cool. Bye, everybody. There we go.